This is a revision series of embryology. So first one, here we start with gametogenesis. So gametogenesis is nothing but here we have both spermatogenesis and oogenesis, that is formation of male and female gametes, right? So let us understand. So the basic timeline is, first we have the pre-embryonic period. So 0 to 2 weeks is known as pre-embryonic period, and the exact embryonic period is from 3rd week to 8th week, and the fetal period is from 9th week to till the baby is born. So what is primordial germ cell? Actually, the key to form all these cells is our primordial germ cell. It is a pluripotent cell. It's synthesized mainly by our epiblasts in our 2nd week. This is important. Synthesized by epiblasts in 2nd week, and from the epiblast it migrate to the endodermal wall yolk sac in the 4th week. So 2nd week it is formed, and the 4th week it is traveling to the yolk sac, and by the 5th week it finally reaches the gonads, where it needs to form the ovary. In case of female we need to form ovum, in case of male we need to form sperm. So all those are formed in gonads itself, so they need to reach the gonads by 5th week. So 2nd week they are in epiblast means forming, and 4th week they are reaching the endodermal wall of yolk sac, and by 5th week they are finally reaching into the gonads. So sometimes what happens is that this primordial germ cell may also reach to some abnormal areas. Actually they need to reach gonads, right? But sometimes they may reach the neck region. If they are reaching the neck, then it is known as Craniopharyngeal Teratoma. So what is teratoma actually? Teratoma is nothing but a defect in which it is formed by 3 layers of the germ cell, that is both all 3, epiderm, ectoderm, endoderm, mesoderm, right? All 3 are involved in case of teratoma. So here this primordial germ cell reaching in the neck region can cause craniopharyngeal teratoma, and sometimes they may also migrate to the region between the sacrum and coccyx. Then the name in this only, that is sacrococcygeal teratoma. So actually why teratoma? Teratoma is nothing but because of pluripotency of the primordial germ cells, right? So here examples are like sacrococcygeal teratoma and craniopharyngeal teratoma, as we discussed. So PGC are nothing but pluripotent cells, that is the primordial germ cell is a pluripotent, which is very important. So let us understand the terms like what is pluripotent, totipotent, all those things. So totipotency is nothing but the ability to form an entire embryo and also extra embryonic tissue up to the 8 cell stage. Every cell is totipotent. So totipotent is nothing but we can form entire embryo from that. So we know 8 cell stage, up to the 8 cell stage, each and every cell is totipotent, that each cell can also form a complete embryo again. So what is pluripotency? As we said, this primordial germ cells are pluripotent, that is ability to form all the germ layers, that is all nothing but we are only having 3 germ layers, that is our ectoderm, mesoderm, endoderm, right? So ability to form all the germ layers is nothing but pluripotency. So here the primordial germ cell, that's why it is causing teratomas, because it is having ability to form all the germ cells. Next, coming to multipotency, it is nothing but it can form more than one category, as the name you can multipotent, right? Here we have hematopoietic stem cells, because these are the cells which can form blood cells, lymphocytes, monocytes. All those cells are formed from the hematopoietic cells. So these are multipotent. Next, oligopotent is that from one category of the cell, the vascular stem cells are like only from one category, forming only one category of cells is oligopotency. So here the example we are having vascular stem cells. Next, what is unipotent cells? Unipotent means, as the name you can understand, only one type of cells they can form. So this is unipotent cell. Here we are having liver cell as an example, which is a unipotent cell. So now let us take the difference of mitosis and meiosis to understand all this progress, because embryology is not all it's all about cells, because from the single cell zygote only of course our whole human body is produced. So we need to understand about the cells, their process of cell cycle, etc. So here we have the mitosis and meiosis. So mitosis mainly takes place in normal body cells, but meiosis we know it takes place in germ cells, gonads, right? So here the chromosome division pattern will be like 46 is divided again into 2 daughter cells having 46. So this is equivalent division. But coming to meiosis, we know it is reductional division, because the 46 chromosomes are finally reduced into, and in meiosis there are 4 daughter cells which are reduced, that is first they are diploid but later they become haploid. But here they are diploid in the beginning and also at the ending they are diploid, but here they are haploid. So this is known as reductional division. And remember that crossing over doesn't take in the mitosis, right? As crossing over doesn't take, the daughter cells are same like the parents, but in the meiosis we know crossing over takes place, so the daughter cells are not resembling the parents. Now coming to meiosis. So meiosis is taking place two times. Yeah, we know meiosis 1 and meiosis 2. And remember that in meiosis 1 prophase we are having diplotin, zygotin, all those phases, right? And the prophase, this phase is common in any case, that is prophase first, next metaphase, anaphase, telophase. So just to remember that. Next coming to oogenesis. So what is oogenesis? Is nothing but the formation of an ovum. So how it takes place, let us understand in a detail. So primordial germ cells first reach the ovary in the 5th week, as we discussed that PGC reach the gonads by 5th week, so in ovary also 5th week. So all the PGC cells are converted into oogonium by 3rd month. So directly they are jumping from 5th week to the 3rd month. By the 3rd month the oogonium is formed. So all the oogonium next converted into primary oocyte by 5 to 7 months, that is in this range 5 to 7 months you can find out the primary oocyte. So as we said, the germ cells should undergo meiosis. So the primary oocyte will undergo meiosis 1 and completes only prophase, but go to resting phase. So it completes the prophase and gets arrested, that is go to resting phase, that is a diplotin phase, and remain in the resting phase until the puberty. So next after puberty, what happens is the primary oocytes completes its meiosis 1 and goes to secondary oocyte and forming a first polar body, so formed due to, so this is all process taking until puberty, but at the puberty what happens is one polar body will be formed, and it is formed due to unequal division of cytoplasm. That is, in the polar body there is no cytoplasm, but in our oocyte, secondary oocyte, there is full cytoplasm. So this phase also undergoes meiosis 2 and completes the prophase and metaphase, and again it arrests in the metaphase after 24 hours. If the sperm will come, then the secondary oocyte completes the meiosis 2 and forms a ovum, and again forms a second and third, fourth polar bodies, right, like that. But if the sperm is not present, then it forms as corpus luteum, yellow substance we know persists for 14 days, and next it shrinks and forms a white scar. It is also known as corpus albicans. So this is what taking place in the case of oogenesis. Now let us see the spermatogenesis. So here also we have primordial germ cell reaching the testis in the 5th week, same like there, yeah, but it will remain dormant until the puberty. This is important. Here it is in a resting phase until puberty in our prophase, but here the male cells are like they remain dormant until the puberty, and at the puberty this primordial germ cell will undergo into convert into spermatogonium. But there the oogonium is formed in the case of 3rd month only, right? So next, the spermatogonium undergoes 5 to 7 cycles of division to form primary spermatocyte. So next, this primary spermatocyte undergoes meiosis 1 and forms secondary spermatocyte, that also 2 secondary spermatocytes. So that is nothing but you can see secondary spermatocyte. So the secondary spermatocyte in turn undergoes meiosis to form 4 spermatids, you can see. So later it undergoes spermiogenesis process, that is nothing but the process of rearrangement of organelles and the shedding of excess cytoplasm, formation of tail, acrosomal cap. So all this process takes place in forming spermatozoa. So what is the process of spermiogenesis? Is nothing but the spermatid is converting into spermatozoa, that is spermiogenesis. Next, spermatogonium undergoes 5 to 7 cycles of division to form primary spermatocytes. So one spermatogonium is forming 64 to 512 spermatozoa, that is one spermatocyte is equal to forming 4 spermatids. Now let us understand some difference between spermatogenesis and oogenesis. So spermatogenesis is the process which is starting only after the puberty, and here the process is beginning in the intrauterine life itself, right? And here the polar body is absent and here the polar body is present. Here the only one spermatocyte can produce 4 spermatids, but here one oocyte can produce only 1 ovum. So the spermatogenesis is completed in 74 days, greater than 64 days and 60 days. So according to options we need to choose it. And the most fertile period in a woman is about 3 days and 2 days before ovulation, or 1 day after ovulation. So because sperms are viable only for 48 hours and the secondary oocyte in the metaphase is arrest and it is waiting only for 24 hours. So this is the reason. And next coming to LH surge. LH surge occurs every 36 hours before the ovulation, and there is LH peak when there is 12 hours before the ovulation or at the rate of ovulation. So what are the one-liner bits here? That is, in male, spermatogenesis takes place in seminiferous tubules, and maturation of sperm mainly occurs in our epididymis, and capacitation of the sperm occurs in a female genital tract, that is about for 7 hours. Okay, this is important. And from which we are having some important points like the part of embryo which exerts a morphogenetic stimulus To the adjacent parts is known as organizer, right? And there is some mutation, gastrulation, there is gastrulation in a embryonic period. That is, what is embryonic period, if you remember? That is from third to 8th week is an embryonic period, right? So embryonic period is the time where the gastrulation takes place and the Leydig cells are forming testosterone, right? And the corpus luteum persists about 2 weeks. So that is important, as we are discussed here, that the corpus luteum persists for 14 days, that is 14, so 2 weeks, right? Done. So these are all important points and bits related to our revision of our gametogenesis. Revision session. So now we are in the first week of development. So what happens in first week? So we know fertilization. It is nothing but taking place in the first week, or that is fertilization taking place mainly in the ampulla of the fallopian tube. So this everyone knows, it's an important point. And we know there is outer zona pellucida layer, which is a membrane. It is mainly preventing the implantation and also polyspermy. And the sperm mainly binds to the zona pellucida first and triggers the release of acrosome enzymes, which are like acrosin, acid phosphatase, hyaluronidase. So all these enzymes are creating a reaction that is nothing but known as acrosomal reaction. We are here, right? So next, the sperm penetrates the zona pellucida and the cortical granules are released and its secretion, that is called as cortical reaction, and the cortical enzymes are reaching the zona. Now the zona is changed, that it is permeable for the sperms, and this is now called zona reaction. So now there is fusion of male and female pronucleus and forming a zygote. This is undergoing the first cleavage forming 2-cell stage, and second cleavage forming 4-cell stage, and third cleavage forming 8-cell stage, and finally undergoing the compaction, that is up to 2 cells to 8 cells stage. The cells are arranged mainly in the loose form, but after 8 cell stage the cells are compaction, that is they are arranged in a tight manner. So what are the reactions mainly taking place when fertilization taking place in order? That is, first the acrosomal reaction takes place, next the cortical reaction takes place, next zona. So acrosomal, cortical and zona. So if we understand this diagram over here, we can get a nice idea over this. So this is the zona pellucida layer. This is the first polar body, and this is a compaction egg plasma membrane. And this is attachment A, and B is binding, C is acrosomal reaction, these are the cortical granules, D is the penetration, E is the fusion and blocking polyspermy, and this is a pre-Wittline space. So that's it. Now we are in the implantation. So implantation is nothing but the 8-cell stage is differentiating into 16-cell stage, and this is called morilla. This is implantation we are going to see it further. First I said till 8-cell stage after compaction, right? So what happens is now the 8-cell stage also undergoes differentiation process and form the 16-cell stage. So the 16-cell stage is called morilla, and this morilla is having cavitation, that is forming cavity in them. Then you know, if any cell with a cavity, it is known as cyst. Here it is known as blastocyst, and the blastocyst undergoes the process of hatching, and the zona pellucida now disappears. It disappears on the 5th day and the implantation takes place. So this is the process of implantation. Next, coming to time and site of events. So fertilization mainly takes place within 12 to 24 hours because for example at the 14th day ovulation takes place according to female menstrual cycle. So at the 14th day when the ovulation takes place, the ovum is only staying in the ampulla only for 24 hours. So within the 24 hours even the sperm need to be there, then only fertilization can take place, right? So 1 day after fertilization we can form the 2-cell stage, and 3rd day after fertilization there is 18 to 16 cell stage, that is a morilla. So 4th day after fertilization there is 32 cell stage, that is advanced morilla, which is entering the uterine cavity and floating over there. So the uterine fluid enters this advanced morilla, that is 32 cell stage, and forming a blastocyst. So at the time of formation of blastocyst, it contains mainly 58 cells in them. Among them 53 cells are forming outer cell mass. So what is outer cell mass? It is made up of 53 cells, that is our trophoblast. We know outer cell mass is forming trophoblast, which future forms placenta. And 5 cells are only taking place to form the inner cell mass, which is our embryoblast. So embryoblast, we know it is forming the embryo proper. And next, blastocyst is formed on 4.5 days after fertilization. This is important. That is complete advanced blastocyst. Okay, so 5th day after fertilization the zona pellucida gets disappeared, and the 6th and 7th day after fertilization implantation takes place. So 6th and 7th day may be implantation taking place. What is ectopic pregnancy? So ectopic pregnancy is nothing but the most common site. We can see this in the fallopian tube, and nothing but there the implantation takes place in the fallopian tube, and the syncytial trophoblast is not well developed in case of ectopic pregnancy. So that is the problem why there is low level of HCG in this ectopic pregnancy, because we know HCG is produced by syncytial trophoblast. So if syncytial trophoblast is only not well developed, then of course HCG also will be very low. And what are the similarities? Like it would look similar to our appendicitis and termination of pregnancy. So what is the treatment we need to do is we need to stop pregnancy, because if we continue this pregnancy, those fallopian tubes may damage. It will lead to tubular rupture. So if not treated, so we need to urgently terminate the pregnancy. So this is all about first week of development, and now let us continue about the second week of development. So this is nothing but the development of embryoblasts and trophoblasts, how they'll form. So as we discussed, there is inner cell mass, which is mainly forming our embryoblast, which also forms two layers, that is columnar cells and cuboidal cells. So columnar cells is the first layer that is forming the epiblast. It is formed on the dorsal end and encloses mainly the amniotic cavity, because for example this is epiblast, here upon we have amniotic cavity, and here is hypoblast, below we have our yolk sac. So cuboidal cells are hypoblast cells forming on the ventral end, and they are mainly forming exocoelomic membrane, also known as Hughes membrane. So it is flattened cell and encloses the primary yolk sac. So remember that the double bleb sign is seen on the ultrasound mainly in the second week. Double bleb means nothing but our yolk sac and amniotic cavity can be seen in the ultrasound if we check it. So this is nothing but double bleb sign, which is important. Next, outer cell mass. Outer cell mass, as I said, it forms the trophoblast. Inner cell mass form the embryoblast. So on the 8th day, the trophoblast is mainly divided again further into two layers. That is a syncytial trophoblast and cytotrophoblast. So the cytotrophoblasts are having the cells which are with single nucleus, and they are well defined membrane, and with a well defined membrane. And coming to syncytial trophoblasts, the cells are multinucleated here, and there is no well defined membrane seen in this syncytial trophoblast. So next, formation of uteroplacental and fetoplacental circulation. So how this forms? It's mainly by lacuna. So lacuna are formed in the cytotrophoblast, and the lacuna are filled with the blood, and also they erode the endometrium and the vessels. So because of this, now the mother blood easily enters into the fetus, right? So the uterine or maternal vessels blood is filled in the lacuna. So through diffusion, this reaches the inner cell mass and supplies the inner cell mass, establishing a uteroplacental circulation. So there's a question like uteroplacental circulation takes place on which day? Then the best answer would be 15th day greater than 12th day. And next, the uteroplacental circulation will be replaced by fetoplacental circulation later. So this fetoplacental circulation is also like having two answers. The better one is 17th day greater than 22nd day. And the heart tube is also formed, which maintains the circulation. We know this, right? So next, coming to chorion and chorionic villi formation, that is extraembryonic mesoderm. So extraembryonic mesoderm is present between the inner cell mass and outer cell mass. That is, it is derived mainly from the primary yolk sac greater than epiblast and hypoblast. So chorion is formed by three sections, that is extraembryonic mesoderm, cytotrophoblast and syncytial trophoblast. Next, coming to the cytotrophoblast, it projects into the syncytial trophoblast forming the primary chorionic villi. So extraembryonic mesoderm projects into this primary villi forming the secondary chorionic villi later. So what is this? We will understand by this diagram. So you can see this is amniotic cavity, this is extraembryonic mesoderm, this is epiblast, this is hypoblast, this is cytotrophoblast, this is primary yolk sac, syncytial trophoblast, and the blood filled with the lacunae, and this they come out like this and forming a primary villi. Next, coming to formation of secondary yolk sac and umbilical cord. So the small cavity is formed in the extraembryonic mesoderm, which joins to form a larger cavity. And the undivided part of the extraembryonic mesoderm, which is mainly cranial to the amniotic cavity, forms a connecting stalk which becomes future umbilical cord. So cavity in the extraembryonic mesoderm is divided into splanchnopluric layer, that is the inner layer of the extraembryonic mesoderm surrounding the yolk sac. So this splanchnopluric layer makes the yolk sac smaller in size, and it converts it into the secondary layer, secondary yolk sac. So the somatic layer of the extraembryonic mesoderm is remaining same like extraembryonic mesoderm, and the amniotic cavity towards the inner surface is mainly lined by the amniogenic cells, and the outer surface of this amniotic cavity is lined by the somatic layer of extraembryonic mesoderm. Next, coming to the summary of second week. So we are completed up to development up to second week. So what is a summary is nothing but by this time 2 layers of inner cell mass are formed, that is epiblast, hypoblast, that is columnar and cuboidal cells, and 2 layers of outer cell mass is formed, that is cytotrophoblast and syncytiotrophoblasts with lacunae with primary secondary villi chorionic villi, right? And amnion and chorion are formed, that is 2 layers of extraembryonic mesoderm, the somatic layer and splanchnopluric layer are formed, and now yolk sac is divided into the primary and secondary yolk sac, right? So that secondary yolk sac only later converts into chorionic cavity. So now let us see the details about human chorionic gonadotropic, that is HCG. So clinically it is correlated with that HCG initially is secreted by our syncytiotrophoblasts. So HCG mainly induces the progesterone to maintain the pregnancy, which is important. So HCG assay, that is on 8th day of maternal blood we can find the HCG, and on the 10th day we can find out in the urine, menstrual urine. This is important. And HCG assay can be done on the first day of the missed period, that is early morning sample is required to find out the HCG, that too in the first day of missed period. So very low HCG conditions can be seen in cases like ectopic pregnancy, as we discussed. Because of low HCG means low syncytiotrophoblasts is not formed well, so there is low HCG levels in ectopic pregnancy, also in spontaneous abortions. And coming to high HCG conditions is seen in multiple pregnancy, molar pregnancy, and also in the gestational trophoblastic neoplasia. So these are all the examples. So by this we completed this revision series. Next, continue with the next one. To channel. Now we are up in third week and placental development. It is chapter 5 and chapter 6 and are in the and embryology. So mainly in this third week what happens is gastrulation takes place and also neurulation. So what this means? Gastrulation means formation of 3 layers. Now till now we know the bilaminar disc, that is epiblast and hypoblast. Now these are converting into trilaminar disc by process known as gastrulation. So now here the process is called gastrulation, and the embryo is now called as gastrula. So this gastrula mainly contains ectoderm, endoderm and the mesoderm. So how they are formed, all those things let us see. And even in the third week neurulation takes place, that is the formation of neural tube. So gastrulation takes place at the day 15 of an embryo. So we have a procaudal plate which is columnar hypoblast cells form the procaudal plate, columnar hypoblast cells. Here you can see this is epiblast, this is a hypoblast. We know epiblast cells are columnar, hypoblast cells are cuboidal, and this towards this hypoblast cells only some of them are like elongated, and you can see this is forming procaudal plate which is mainly present towards the head end, and these are columnar hypoblast cells. So which are mainly forming buccopharyngeal membrane, and this buccopharyngeal membrane further ruptures and forms a oral cavity. So we know at the head end only we are having oral cavity. Next at the tail end we are having a primitive streak which is formed by the proliferation of epiblast cells, and the primitive streak indicates the beginning of the gastrulation process. So primitive streak mainly it is formed at the end of the second week or during the beginning of the third week, and it persists at the tail end, and sometimes a sacrococcygeal teratoma may form, and or it is also due to abnormality in the migration of primordial germ cells in our previous one if you remember. So first germ cell layer to develop. As I said, gastrulation means 3 layers. So what is the first germ layer? It is nothing but endoderm. So endoderm forms first. It is derived from the epiblast itself, and the endoderm replaces the hypoblast, that is a lower layer. And next the mesoderm is the second germ layer. It is derived from the primitive streak and derived from the epiblast, because primitive streak is in turn formed from the epiblast, right? So last germ layer is the ectoderm, that is uppermost, the remnants of the epiblast after formation of the intraembryonic mesoderm. So the remaining part forms the ectoderm. So all germ layers are mainly derived from epiblast, of course, greater than yolk sac. Sometime according to options we need to say it. So from primitive streak stimulates formation of neural tube itself. It's a midline notochord. So the midline notochord is also part of mesoderm, but forming a midline structure. From this only the primitive streak stimulates and now forms a neural tube, right? So the structures derived from notochord are nucleus pulposus and apical ligaments of dens. And there is nothing but the remnants in adult are nucleus pulposus mainly, and also apical ligaments of dens. But the greater one is the nucleus pulposus. And next, notochord disappears later. in a later age. So if persists sometimes, it may form a tumor-like substance known as chordoma. It is a congenital tuber, and it is having some cells known as physiferous cells which are present in the chordoma. So these physiferous cells are large vacuolated cytoplasm, and the nucleus is mainly towards the periphery. We are here. Now next, subdivisions of intraembryonic mesoderm. So intraembryonic mesoderm is nothing but now the form mesoderm between the ectoderm and endoderm is the intraembryonic mesoderm. It is divided into paraxial mesoderm and intermediate mesoderm and lateral mesoderm. In this diagram we can understand, for example, this is a notochord. Here will lie the paraxial mesoderm, here lies the intermediate mesoderm, and more laterally lies the lateral mesoderm. Right, so the paraxial mesoderm is present on either side of the notochord and mainly forms the somites. What are the somites? Let us see later. Okay, an intermediate mesoderm forms the urogenital system mainly, and the lateral mesoderm, due to its cavities, again further divided into parts like 3A and 3B. So the 3A is nothing but splanchno-puric layer. That is forming visceral, cardiac muscle, smooth muscle, and also it forms splanchno-puric layer of the body cavities. And we are having the 3B, that is somatic layer, which mainly forms appendicular skeleton of both upper limb and lower limb bones, and also somatic layer of the body cavities. And next, coming to somites. So somites are nothing but which are present in our paraxial mesoderm. So this here the paraxial mesoderm. The somites are like divided into sclerotome and dermomyotome, and dermomyotome is further divided into dermatome and also myotome. This you need to understand. So this is together somite. So somite only sclerotome, dermamyotome. Dermamyotome is again divided into dermatome and myotome. Right. So somites, first pair is present mainly in the occipital region. At the 20th day you can see this, and each day 3 pairs are getting added to it. So now we can count on 21st day 4 pairs are present, on 22nd day 7 pairs are present. Like that on 35th day, that is about 5th week, there are 42 to 44 pairs of somites are present. So how the order is nothing but the order of appearance of somites is craniocaudal sequence, that is from head end to the tail end, that is occipital, first cervical, next thoracic, next follows lumbar, sacral and coxial. So you can understand this from cranial to caudal end. And next, after the 5th week, some somites form from the occipital and coxial region get disappear. So what happens is, so now total only 37 pairs approximately are present after the 5th week. Next, sclerotome. So sclerotome forms the axial skeleton, that is vertebra. So sclerotome we have seen, right? This forms the axial skeleton or the vertebra. And next we have what is a sclerotome. Sclerotome is divided into again ventral, lateral, dorsal parts. So let us understand over here. Here I wrote that sclerotome, it go around the notochord like this, and also in the go around the notochord and neural tube and fuse to the opposite side. For example, if it is started rounding from this, it like this rounds, and from here also, and fuse at the opposite side. So this is nothing but now the sclerotome is forming a structure like this, and ventral, it is ventral sclerotome, it is lateral sclerotome, it is dorsal sclerotome, right? So ventral sclerotome is forming mainly the body of the vertebra, which fails to fuse, then it may form hemivertebra. And dorsal, it is forming the spine of vertebra, and ventral is forming the lateral is forming the vertebral arch. So if the dorsal sclerotome fails to fuse, then it is forming the spina bifida anomalous. So spina bifida anomalous are divided into spina bifida occulta, meningocele, myelocele. So let us see those things. So spina bifida occulta is dorsal sclerotome fails to fuse. Yeah, tuft of the hair is present over the defect. It looks hairy like structure. And meningocele, dorsal sclerotome again fails to fuse, and meninges protrude over the defect. And myelocele, dorsal sclerotome fails to fuse, and the protruded meninges contain the spinal cord in them. Next, dermamyotome is subdivided into dermatome and myotome, and the myotome is having skeletal muscle of the forming the skeletal muscle of the body, and the dermatome forms the dermal layer of the skin. So this we need to remember. In the myotome, as the name you can understand, like muscles, forming muscles, derma, forming dermal layer. So in the name only you can understand these things. So note, there are like some important points, like smooth muscle which is present over the dorsal aorta is mainly made up of splanchnopubic layer of the lateral mesoderm greater than paraxial mesoderm. Because this lateral mesoderm, okay, greater than, that's it. Smooth muscle over pharyngeal arch arteries, neural crest cells, and smooth muscle in iris, neuroectoderm. So pharyngeal arch arteries are of neural crest cells, and smooth muscle in iris, neuroectoderm. Smooth muscle over sweat and mammary glands are of surface ectoderm. So in humerus, we are having the somatic layer of lateral mesoderm. What they are saying, somatic layer of lateral mesoderm. Lateral mesoderm, we know it is divided in splanchnopubic somatic. So somatic layer forms humerus, and biceps muscle myotome, because it's a muscle, right? Myotome. That's the somites of paraxial mesoderm. Why? Because paraxial mesoderm is only divided, right? So that's it. Next we are seeing angiogenesis and hematopoiesis. So hematopoiesis, first it is nothing but the synthesis of blood vessels. In the name you can understand. So here the mesoderm is nothing but differentiating into the blood vessel, and territory villi is also formed by the end of third week. Why they added this point here? Because in territory villi blood vessels are present, right? Territory villi is formed by the end of the third week, or also we can say on the 21st day. So what the territory villi contains? It contains blood vessels, somatic layer of extraembryonic mesoderm, cytotrophoblasts, syncytial trophoblasts, splanchnopubic layer of extraembryonic mesoderm, which differentiate. That's it from here. How it forms is, first is splanchnopubic layer of extraembryonic mesoderm differentiate first, and next then the somatic layer of extraembryonic mesoderm forms, and next the intraembryonic mesoderm may follow it. So hematopoiesis. Hematopoiesis, in the name you can understand, synthesis of blood cells. Third week splanchnopubic layer of extraembryonic mesoderm only lead to hematopoiesis. So after the fifth week, the organs which have this function is liver, thymus, spleen and bone marrow. But liver, remember, up to only 7th month it has a function of hematopoiesis. And we know the major part in all the adult life is of hematopoiesis is done by our bone marrow. Next, coming to the development of placenta. So we know placenta is having maternal component, fetal component. In the maternal component we have decidua basalis, and in the fetal component we have our villus chorion. And decidua means what? That is the uterine endometrium during the pregnancy, under the corpus luteum of pregnancy because of progesterone action. So this is known as decidua. That's it. It's endometrium only, but the name is changed during the pregnancy. So the decidua is further divided in three types, like decidua basalis, capsularis, parietalis. According to its name only we can understand. Basalis means in the base, that is at where the uterus get implanted to the female endometrium. That site is decidua basalis. Between the fetus and the myometrium. So where it is present? Between the fetus and the myometrium. And decidua capsularis is between the uterine cavity and the fetus, because it's like a capsule. It forms around outside. And parietalis is rest of the decidua. So all over the rest part of the decidua is covered by parietalis. And the first chorionic villi is made up of cytotrophoblasts and syncytial trophoblasts. Second chorionic villi is made up of cytotrophoblasts, syncytial trophoblasts and mesoderm. Third chorionic villi, cytotrophoblasts, syncytial trophoblasts and also blood vessels. It's the 21st day tertiary chorionic villi is formed. So that's it until now. Of embryology. So now here we are with a head and neck. So by fourth week what happens is the neural crest cells mainly start migrating to form the future neck, which forms the pharyngeal arches. So first let us see this diagram so that we can get an idea what is this pharyngeal arches, etc. So developing embryo, you can see it's a head bulge where head forms in future. This is a cardiac bulge, and between you can see here the stomatium is present, which converts into future mouth. And yeah, this is a head. Cardiac bulge is now between. So here the pharyngeal arches are formed in a ventral side, you can see. So by the fourth week what happens is the neural crest cells start migrating here to form the future neck, so which forms the pharyngeal arches. So what are pharyngeal arches? They are mainly mesenchymal condensation. Mesenchyme is derived from neural crest cells and also from the paraxial lateral mesoderm. If you remember that paraxial and lateral mesoderm. So from paraxial and also from lateral mesoderm and also from neural crest cells, this mesenchyme is derived. And there are 5 pharyngeal arches mainly, but there is also 6th one here. The 5th one disappears later. So only 1, 2, 3, 4 and 6. The 5th one disappears later. So the outer layer is ectoderm and the inner layer is an endoderm. In middle we having the mesoderm. In the mesoderm itself we have this pharyngeal arches, and outer layer ectoderm forms the clefts and inner layer endoderm forms a pouches. So the spaces between the ectoderm form the clefts. There are about 4 clefts, and the spaces between the endoderm form pouches. So there are about 4 pouches. So there are 4 pouches, 4 clefts, and total 5 arches. This is important. And what are the derivatives of arches mainly? So the components of pharyngeal arches are like, in a mesoderm you can find out the cartilage, the muscle, nerve, artery. So all these are present: cartilage, muscle, nerve, artery. You can see a aortic arch over there in the place of artery, right? So now let us see the arches in detail, that what time means in which arch what kind of cartilage is present, what kind of nerve is present, what kind of muscle is present, what kind of artery is present. So let's start with cartilage. So in the first arch we have our Meckel's cartilage, which mainly forms malleus. So it's cartilage, right? It converts into bone by endochondral ossification. So Meckel's cartilage forms malleus, incus, small bones of our ear, and it forms anterior ligament of malleolus and sphenomandibular ligament, maxilla, mandible, zygomatic, sphenoid, and temporal bone. So all these are formed from first arch cartilage, Meckel's cartilage. And the nerve which is in the first arch is a mandibular nerve, and the muscle is a muscle of mastication. So a muscle of mastication, temporalis, mesoscler, medial and lateral pterygoid, anterior belly of digastric, mylohyoid, tensor palatini, and tensor tympani. And next coming to the artery. The artery, here we have the maxillary artery. So maxillary nerve and maxillary artery. And the second arch mainly having a Richard cartilage, which is, you can remember like 5S, that is styloid process, stylohyoid ligament, small horn of hyoid, superior surface of hyoid. And the nerve is the facial nerve or the 7th nerve, and the muscles are muscles of face, posterior belly of digastric, stylohyoid, auricular muscle, and also stapedius. And the artery is here stapedial artery. And next coming to the third arch. Third arch is having inferior surface of hyoid, greater horn of hyoid, and the nerve is glossopharyngeal nerve or the 9th nerve, and the muscle is stylopharyngeus, and the artery is again common carotid artery, internal and external carotid artery. Next coming to the fourth arch. Fourth arch is mainly having a cartilage of larynx except epiglottis. Even the sixth arch also having a cartilage of larynx but except epiglottis. So here again the nerves are superior laryngeal nerve in the case of 5th arch, and 6th arch we are having our recurrent laryngeal nerve and the superior laryngeal. And the muscles of 5th arch are muscles of pharynx, palate, cricoid. Muscles of 6th arch are larynx except cricoid, because cricoid is from our 4th arch itself. Right, so this is a 4th arch, this is a 6th arch. And here we have right side we are having right subclavian artery, and left side we are having arch of aorta. And in the case of our 6th arch we are having pulmonary artery, distal part on left side, on left side distal part, pulmonary artery distal part, and on left side we are having the ductus arteriosus. And as we said, except epiglottis, then from where epiglottis forms, it is mainly derived from the mesoderm of hypobrachial eminence of 4th arch. Next coming to Tucker-Collins and Perry-Robinson syndrome. Let us see these syndromes. What is Tucker-Collins syndrome? Here the first arch derivatives fail to differentiate. And clinical features will be having a facial symmetry, and we have a colobome of lower eyelid and external ear anomalous. Next coming to the Pierce-Robin syndrome. Here only the mandible is not well developed, and clinical features are like hypoplastic mandible cause posteriorly displaced trunk, that is glossoptosis, that obstructs the palate fusion and causes the cleft palate. So in Pierce-Robin syndrome you can see cleft palate, you can see glossoptosis, and also you can see hypoplastic mandible. So these three are the features of Pierce-Robin syndrome. Next, let us move on to derivatives of endodermal pharyngeal pouches. So we have completed arches. Now we are going to do pouches. So we know there are 4 pouches, right? So the first pouch from middle ear eustachian tube, second pouch from tonsillar fossa, tonsillar epithelium, third pouch from ventral thymus, dorsal parathyroid, and becomes inferior parathyroid. And the fourth pouch forms as ventral ultimobrachial body parafollicular cells of thyroid, and the dorsal forms parathyroid 4 superior parathyroid. 4 means superior parathyroid, 3 means inferior parathyroid. Now let us see the parafollicular cells, how the parafollicular cells are derived from. They are mainly derived from the neural crest cells, that is ultimobrachial body. We can say according to bits like parafollicular cells are mainly derived from neural crest cells greater than ultimobrachial body. Next, coming to clinical correlation like DeJoy syndrome, where there is 22q11 deletion, and the third pouch is failed to differentiate, and the thymus and inferior parathyroid are here poorly developed. Next, coming to ectodermal cleft. Here there are clefts means there are 4 clefts. So the first cleft forms extraordinary meatus. The growth of second arch fuses with the sixth arch to form a cervical sinus, which is obliterating later. So if the cervical sinus persists in some persons, then it may cause bronchial sinus. Anywhere on the anterior border of sternocleidomastoid we can see this, and usually found below the angle of mandible. So cervical sinus is obliterated other 5 clefts. So in ectodermal cleft we only have 1 cleft because all other clefts are obliterated by a cervical sinus. Next, let us see the development of tongue. So how tongue is formed. Epithelium of tongue is anterior to that by surface ectoderm, because we have discussed already the structures which are exposed outside are lined by surface ectoderm, and the posterior 1/3 is lined by endoderm. So the muscles of the tongue all are derived from the occipital somites except the palatoglossal muscle, because this is derived from the 4th arch. This is important. Next, muscles of the tongue we have styloglossus, palatoglossus, hypoglossus, genioglossus. We have already studied this in our simple anatomy, right? And in the floor of first arch we are having 3 mesodermal swellings, 2 lingual swellings, and tubercular impar. This is mainly forming the anterior 2/3 of the tongue, that is 3 mesodermal swellings, 2 lingual swellings, and the tubercular impar together form the anterior 2/3 of the tongue. And the 2 to 4 arches form the hypobronchial eminence. So the hypobronchial eminence have in the 3rd arch its mesoderm form the posterior 1/3 of the tongue, and the 4th arch posterior most part of the tongue, the 4th arch of the hypobronchial eminence form the posterior most part of the tongue along with the epiglottis. Next, the 2nd arch mesoderm from the hypobronchial eminence is not contributing to tongue development because the mesoderm of 3rd arch is overgrowing and it fuses with the anterior 2/3 of the tongue. So each arch have post-traumatic nerve, and let us see the post-traumatic and pre-traumatic nerve. So what is pre-traumatic nerve? But remember that pre-traumatic nerve is only present to the 1st and 2nd arch, where pre-traumatic nerve means the lower arch is giving the branches to the higher arch. This is known as pre-traumatic nerve. Now, first arch is chorda tympani. It is giving pre-traumatic nerve. It's chorda tympani, and the first arch post-traumatic nerve is mandibular nerve. Second arch post-traumatic nerve is facial nerve, and the second arch pre-traumatic nerve is tympanic branch of glossopharyngeal nerve. Third arch post-traumatic is glossopharyngeal nerve. From third arch, remember there are no pre-traumatic nerves. Next, coming to development of pituitary. So pituitary, from stomatium we get a blind pouch called as Rathke's pouch, which is also lined by surface ectoderm derivative. So it gives rise to pars tuberalis, pars intermedia, and also adenohypophysis. But the neuroectodermal derivation, that is diencephalon, is downgrowth forming an infundibulum giving rise to pars nervosa. Pars nervosa means posterior pituitary. So neuroectodermal derivative is only posterior dormitory in the case of pituitary. So next, coming to development of face. So in face, the main part, there are 5 processes which are leading to the formation of face. That is our nasal pit. We are having lateral nasal process, frontonasal process, medial nasal process, and from first arch we are having maxillary and mandibular process. So these 5 processes together lead to the formation of face. So let us see what happens. First, the 2 medial process fuse together, they form the philtrum. So let us see one by one. So first one we'll see lateral nasal process. It forms the ala of the nose, that is the side part of the nose, and the frontonasal process mainly forms the forehead, and the medial nasal process fuse with each other to form the philtrum and also primitive palate. And next, coming to the maxillary process, which give rise to cheeks, lateral part of upper limb, etc. And the mandibular process give rise to the lower lip and also chin. So mainly maxillary process fuse with the medial and lateral nasal process. Sometimes what happens is two medial process fuse, but they fail to fuse, then philtrum doesn't form, and midline cleft upper limb is a thing which is formed. So what is cleft upper limb? That is mainly due to the failure of fusion of maxillary and medial nasal process, or it is also known as hair lip. And oblique facial cleft is the maxillary process fails to fuse with the lateral nasal process, and cleft in the lower lip, that is a failure of lower lip fusion in the mandibular process. So this is all about quick revision, because as we already learned this many times, we know all the concepts, so it's just like a quick revision.